our project aimed to do an unbiased screening of autoreactive T-cell responses in MS patients. As I mentioned before, we know that patients are very heterogeneous, and our lab previously used the bead platform described in my presentation to revisit the classical autoantigen MOG. And we found by coupling paramagnetic beads to MOG and screening our patients for T-cell reactivity against MOG protein, we found that only 50% of patients actually had a T-cell response to MOG. And therefore, we reasoned that there must be other autoantigens that are undiscovered in patients. So we revisited proteins, made a list of 63 candidate autoantigens from the Human Protein Atlas. We coupled these to beads in, I think it was 63 pools, and used a form of ELISpot called Fluorospot and our bead technology to see which pools of bead coupled antigens were targets of autoreactive T-cell responses in MS. We used a Natalizumab treated cohort, as these patients have a higher, the Natalizumab drug itself excludes autoreactive T cells from the central nervous system. So in theory, these patients should have a higher number of circulating autoreactive T cells in their blood. And we found that 4 pools out of the 63 were highly responded to compared to the healthy control group. And these responses were mapped to 4 proteins, and these were FAB-P7, PROC2, RTN3, and SNAP921. We looked in a larger cohort of Natalizumab donors and found this larger cohort to replicate the results in the screening study. And then also we looked in a second validation cohort, which was untreated patients, and found these to be targets still in this second cohort. We also looked at antibody responses to these proteins, and we found that one protein in particular was a common antigen antibody target, but the other 3 weren't, which was quite interesting. We also looked in a mouse model and immunized with complete Freud's adjuvant mice with these 4 candidate autoantigens. And we found that whilst there was no change in the clinical score of the mice, we found that there was infiltration into the brain and spinal cord for these after immunization with these proteins in the mouse model. We also validated our Fluorospot approach with flow cytometry, and so our results were replicated with a different method. And we found that the T-cell responses using flow cytometry found that these proteins were predominantly responded to by CD4 T cells in the patient cohort, and that they mostly produced interferon gamma, but also there was some production of other cytokines such as GM-CSF. And we also found that in MS patients, there was more of a skewing towards the CD4 T cell compartment rather than to the CD8 compartment. And this was specific to the MS group, whereas the healthy group, if there were responses to these antigens, these autoantigens, it was not necessarily CD4, it could also be CD8. We also looked in the untreated cohort and looked at how good a diagnostic test could detecting autoantigen-specific T-cell responses to proteins be in this way using Fluorospot. And we found that we used the receiver operating characteristic, or ROC curve, to do this. And we found that responses to 4 autoantigens, so MOG, MBP, PROC2, and RTN3, looking at responses to these 4 autoantigens in combination gave a ROC value of 0.88, which fits the minimum criteria for use as a diagnostic test. But individually, responses to these autoantigens were not sufficient to predict disease compared to healthy controls. So this does have potential use as a diagnostic test in future, we hope. So the implications for our study are we hope that this platform using autoantigen coupled beads will be used to screen patients for autoreactive T cell responses, either for their inclusion in tolerization trials or in diagnostics. It will also allow us potentially in future to investigate autoreactive T cell responses in an unbiased way. As full-length antigen can be coupled to beads, it's natively processed by the patient's own cellular machinery in a non-HLA dependent manner, and also allows full-length antigen produced quite cheaply in bacteria or in a mammalian system to be washed of contaminants, which can drastically reduce background of assays and increases sensitivity of our approach. And it's also very versatile, so it can be applied in theory to any protein antigen across autoantigen diseases or virology or anything. So we hope that the implications and usefulness of our approach will be much greater than just the MS field.